Okay, so biofertilizers are any substance of biological origin or live organisms or a combination of thereof which can improve the yield, quality or nutrient by improving the nutrient uptake efficiency of the plants and also stress tolerance. So they improve the quality and quantity of uh, the output, the produce, that is uh, fruit or grain or salad. So they help in the nutrient uptake mostly, and they may be one organism or a combination of them, which is called microbial consortium. They were they were known to have beneficial effect on the plants long back. So first commercial biofertilizer was nitrogen, that was launched in 1896, and uh, today the market is around 2-3 billion for biofertilizers. Of these biofertilizers, the this market is uh, this is composed of around 5% of total fertilizer market. So 95% are normal fertilizers and 5% are biofertilizer. But slowly the share of biofertilizers is increasing. And in biofertilizers, most around 80% is rhizobium based. Rhizo So people use the rhizobium bacteria for improving the uh, nitrogen fixation in leguminous crops because they produce the root nodules. They help in production of root nodules. And root nodules of leguminaceae family, they fix the nitrogen from the atmosphere. Okay, so these are biofertilizers, and now we will first study the first class, one class of these biofertilizers, which is blue green algae. So, what are blue green algae? These are bacteria. These are cyanobacteria. So, these bacteria are also called blue green algae, and they many of the blue green algae fix the nitrogen. So, they are also used as nitrogen fixers. Uh, there are other uses of cyanobacteria also for uh, use as a fodder or a protein source or for making this biomass, green biomass for manure preparation or medicinal preparations or dietary protein supplements. So, uh, the cyanobacteria fix the nitrogen, and they are so one class of biofertilizers. So, uh, cyanobacteria are the most abundant group of organisms on the earth. They arrived very at the beginning of the origin of life. So, just after the first prokaryotic life originated on the earth, simultaneously cyanobacteria also originated. Not simultaneously, immediately after that. So, the first life is supposed to origin around 3.5 billion years ago, and the cyanobacteria also maybe few million years later. So the cyanobacteria also said to arise around 3.5 billion years ago. And when did the Earth origin? Origin of Earth is 4.5 billion years ago. So they are found in diverse environment, but mostly in marine water. And if they are to be cultured, then marine water is the good nutrient source. They are generally unicellular and grow in large colonies. Cyanobacteria are composed of broad range of bacteria with different sizes and shapes. Around 150 genera are known. They gave rise to endosymbionts. Endosymbionts mean when these cyanobacteria were engulfed by another bigger prokaryotic cell, they became chloroplast. And this way, the uh, origin of chloroplast happened. So this process is called endosymbiosis. So the chloroplast origin is endosymbiotic. Mitochondria also originated in the same way. Now, what does the cyanobacterial cell contain? It contains numerous inclusion bodies, which contains light harvesting complexes, which contain thylakoid membranes, phycobilisomes, polyphosphate bodies, polyhydroxyalkanoate granules, then carboxysomes, cyanophysin granules. Cyanophysin granules acquire large polypeptides containing approximately the same number of amino acids such as arginine and aspartic acid. They are visible under light microscope and store more nitrogen. So cyanophysin, same cyano word is used. So if you see the pigment of blue green algae, uh, they are chlorophylls, different chlorophyll A, B, then xanthophylls, then C phycoerythrin and C phycocyanin. So C means cyanobacterial. The cyanobacterial phycoerythrin and phycocyanin. C phycoerythrin, C phycocyanin. Now they contain carboxysomes. Carboxysomes means those uh, paracrystalline bodies which contain a lot of rubisco. Rubisco is concentrated in carboxysomes, and some of it is released into the cytoplasm and some of it there itself fixes the carbon dioxide. So Kelvin cycle takes place in carboxysome as well as uh, slightly and to some extent in cytoplasm. Then lipid bodies, DNA containing regions and ribosomes, all these are inclusion bodies in the cyanobacteria. They are supposed to uh, origin in the late Proterozoic or early Cambrian. Uh, I mean, the cyanobacteria were found earlier, but the endosymbiosis happened in the Proterozoic or early Cambrian period. So these are the history of evolution is divided into eras and periods. So there are many uh, periods like Silurian, Devonian, Carboniferous, Cambrian. So the cyanobacteria endosymbiosis happened in Cambrian period. Now the structure, they have similar structure uh, of cell wall. I mean, the outer boundary is similar to bacteria. That is, uh, in bacteria, membrane is there, then cell wall, and then capsid is there. So similarly here also membrane, cell wall, and outer mucilaginous layer, and it contains pigmented lamellae not into plastid. So it is obvious because they they themselves became plastid. So in the plastid you have thylakoids. So Thylakoids are present in the grana in chloroplast, but here the thylakoids are present in the, they are actually thylakoid lamellae. Lamellae means thread like structures. Instead of present in disc form and making grana, these uh, the thylakoids are present in a lamellae form in the cytoplasm of cyanobacteria. And so this C-phycoerythrin and C-phycocyanin, they are only found in blue-green algae. And classification of BGA, chlorococcales, they are, so what are the examples of blue-green algae? Nostoc, Enbina, Tolipothrix, uh, Cytonema, Stigonema, and Oscillatoria. So based on this, the representative cyanobacteria, they are divided into different orders. So the orders are four. Chlorococcales, Chlorococcus based on Chlorococcus. Nostoc gave rise to this order. Nostocales. Oscillatoria gave rise to this order. Oscillatorials and Stigonema. Then, so they fix nitrogen. Most efficient nitrogen fixers are Enbina variabilis, Nostoc linkea, Alocira, Calothrix, Tolipothrix, Cytonema. And they fix around 20 to 25 kg per hectare per year, but uh, even more up to 60 kg per hectare. Now, how they are mass cultured? The mother culture is kept in the fridge at minus 20 degree or minus 80 degree. From there, it is put in the slant. Slant means test tube containing the media. When the media is liquid, the test tube is uh, kept in the slanting position so that the media make a slope on the top. Instead of being at horizontal position, it makes a slope. It is not, the surface is not perpendicular to the tube height, tube length. So the purpose is just to increase the area where you can put your sample. So where you put your sample, it will grow from there. So you have more growth of the initial mother culture. So mother culture is put on the slant test tube media. Then from there it is put in broth. Broth means liquid media where you put this from this slant. When the alga grows, you scrape it and put into the liquid nutrient medium, and you shake it at the proper temperature. After some days, it will grow and your broth is formed. So that broth is then put into more media, and so big you got more broth. From there, it can be shifted to the fermenters. Fermenter are big in size, and you can culture a lot of cyanobacteria or bacteria there. So after the fermentation process is over, you have got all the cyanobacteria good yield. Then you filter it or centrifuge it to separate from media. Then you dry it or remove the moisture. Then you do the packaging in the bags. You can then at 25 degree you can put for seasoning for some time, like, and then quality check and dispatch to farmer. So this can be used for by farmers for this can serve as a seed. So it will, when the farmer spread in the field, there it will spread more and fix the nitrogen more. All the biofertilizers act in that way only, that you spread the live organism. They will grow in the, they will divide and grow in the cell, in the soil, and this way their number will multiply tremendously, and they can then do their action, which may be fixation of nitrogen, mobilization of other nutrients, or solubilization of nutrients. Now, five critical abiotic parameters. So, if you are doing culture of these in the fermenter or in the flask, there are some critical parameters that you need to standardize and maintain, which are pH of the media, then light condition, because they do photosynthesis, so they need light, pH, then nutrient composition, and then carbon dioxide, and uh, temperature. So these conditions you need to maintain, then slowly you learn how to grow them properly. And these some bacteria, some cyanobacteria have been used to get high yield: Arthrospira, chlorella, Hematococcus, and Dunaliella. So next, after you make this, in the similar way you can grow your bacteria also. So after growing the bacteria or cyanobacteria in the bioreactor or fermenter, then you separate it, and then you mix with inert material, carrier material. So that carrier material should have what properties? Carrier material में मिला दोगे तो formulation is ready. That formulation can be sold in the shops. तो वो formulation में क्या होता है? What is there in that formulation? What should be the quality of that carrier material? The carrier material should be non-toxic to animal as well as plants. Should be inert. That means it should not release any chemicals or change the pH of the solution. So it should be inert. Good buffering capacity. If there is a buffering capacity, then it is good. Buffering capacity means if the pH is high, it will reduce the pH. Not high means due to some factor. If suppose you are spraying the cyanobacteria in this, after buying this biofertilizer of cyanobacteria, you spray it in the field, but your field is alkaline. There your cyanobacteria cannot grow. But because this cyanobacteria is there in the inert material, and that inert material has buffering capacity, so the soil alkalinity will affect less, uh, will less affect this bacteria or cyanobacteria. The buffering capacity means to oppose any change in pH. So suppose if the soil pH is 4 and I spread that formulation, so the in the this cyanobacteria formulation that is in the inert material, so that formulation, the pH of that formulation should also change to 4, because whatever is causing the low pH in the soil, this chemical will diffuse into the formulation also, so it will also try to decrease the pH. So if the pH of formulation was 7, then slowly it will become 4. But if there is a buffering capacity in that formulation, in that carrier material, then it will resist that change. It will try to remain at 7 pH. Instead of suppose 4 outside, it will just become 6 or 7. 5 or 6 pH. So buffering capacity is good for action of cyanobacteria. Then it should have good absorption capacity. It should be able to stick to the roots and to the seeds, not roots, to the seeds if we are treating the seeds. Then it should be easy to operate. It should act as a food for initial growth of bacteria. It should be available in the high amount and adequate amount, and it should be cheap, and it should be able to be sterilized by gamma radiation or autoclaving, because if you contaminate your biofertilizer, then you are not giving the proper product to the farmer. So first you sterilize the carrier material, then you add your bacteria into that, then your ready to use formulation is available. What are the example of formulations? Peat, perlite, charcoal, soil aggregates, etc. And starch is also used, and chalk powder is also used. So peat is the most frequent carrier material, and if it is to be used in the soil, then instead of powder, granules are also made, which are 0.5 to 1.55 millimeter. Sterilization of carrier, I told already that carrier material, if it is contaminated, that contamination will spread and it may kill your bacteria also. So it is better to sterilize the carrier material to prevent the contamination. So to sterilize, you can use autoclaving or gamma radiation. Gamma radiation is given at 50 megahertz. Okay, and what are the requirement as per government rules? As per the government requirement, what are the quality control parameters in the biofertilizer? It should be free of contamination. The product name should be given on the packaging requirement. What are the packaging requirement for biofertilizer? The product name should be there, company name should be there, address should be there, crop, suitable crops where it can be used should be written, carrier type is written, batch number, batch code, manufacturing date, expiry date, net quantity, and instruction for use and storage. All this information has to be written. Next, so what are diazotrophs? Diazotrophs are nitrogen fixing bacteria. So this are another. So nitrogen fixing are the most prevalent and most used biofertilizer. Others are slowly coming into the market, which are zinc, calcium, and phosphate and sulfur based biofertilizers. Based means they will degrade the complexes of these chemicals so that these nutrients are solubilized and made available for the plants to be absorbed by the roots. And PSB means phosphate solubilizing bacteria can increase yield 10 to 20%, and nitrogen fixing bacteria also to the same amount. Then market, as I told earlier, around this is expected data in 2016 that by 2022 it will reach to $2.3 billion, out of which rhizobacteria is of maximum 80% share, and then second is of phosphate solubilizing bacteria. Next, diazotroph. What are there in the diazotroph? Means nitrogen fixers. They have this heterocyst. One specialized cell is there which will fix the nitrogen. How it fixes? This nitrogenase enzyme is there, which will, after using the ATP by the formation by photosynthesis process, ATP are produced in the photosynthesis. So that ATP is used for converting nitrogen gas into ammonia through the enzyme nitrogenase, and uh, ammonia is further converted to other chemicals which are glutamine and glutamate. So all these are then transported to the next cells, because here only in this heterocyst nitrogen fixation takes place, which is then the fixed nitrogen means nitrogen is converted into other compounds like ammonium ions, or nitrate, or this glutamine, glutamate. So everything here is nitrogen. You, the conversion of nitrogen gas into any of these things is called nitrogen fixation. Okay, next rhizobium. So we will study different bacteria now, biofertilizers. So first one is rhizobium. So I told earlier there are different species of rhizobium which are suitable for different plants. So Bezrink martinus, Bezrink first isolated and cultivated rhizobium from the nodules of legumes in 1888 and named it Bacillus radicicola. And rhizobium is a genus of gram-negative soil-fixing bacteria. Gram-negative means they have thinner walls. They will not get this gram-positive stain. So they fix the nitrogen, and they are gram-negative bacteria. They are they form endosymbiosis in the root nodules of legumes and parasponia. One plant is also there which is not leguminous, but it also are nodules. Bacteria colonize plant cell within the root nodules, convert atmospheric nitrogen into ammonia using the enzyme nitrogenase, and provide organic nitrogenous compounds such as glutamine, glutamate, urides, etc. So in the same way as the heterocysts do nitrogen fixation in blue-green algae, the initial process is same in the rhizobium. Next, how this root nodules is formed. So this is the this is a transverse section of root. The root have these hairs, and in the hairs, not in the hairs, and you have this clumpy mass which will form the root nodules, and inside this in blue color, this is bacteria. When they are outside, they are called bacteria. When they are present in root nodules, they are called bacteroids. So this is how bacteroids are present into the rhizobium bacteroids inside the root nodules. And the formation of functional nitrogen fixing nodules depends on two highly coordinated processes. One, infection by rhizobia, organogenesis of nodules. Non-nodulating rhizobia. There are other rhizobia also which do not cause the nodulation, which are example are bradyrhizobium on acmeaspon plant. On this plant, they do not form nodules. Robust heterotrophs that can persist in bulk soil thrive in the rhizosphere or colonize roots as endophytes. So the rhizobium can be present in the rhizosphere or in inside root as endophyte. Relative frequency of non-nodulating versus nodulating rhizobia also varies, but non-nodulating genotypes typically dominate and can encompasses 99% of total rhizobial population in the soil. Okay. Next, so how this node formation happens? Node factors such as lipocyto-oligosaccharides are secreted by rhizobia, and they are perceived by the receptors present in the plasma membrane of root cells. So initially, root plant roots secrete cytokines. These chemicals will attract the rhizobium bacteria, and then this rhizobium bacteria releases lipocyto-oligosaccharides that will stimulate the plant root to form root nodules, and the hair is the root hair traps initially this bacteria inside, and then hair nodules formation happens. Now next biofertilizer is azospirillum. It is a free living nitrogen fixing gram negative bacterium closely associated with rhizosphere, isolated from the rhizosphere of many grasses. So this is mostly azospirillum is used mostly in grass family plants like rice, wheat, corn, maize, millets, jawar, bajra. Okay, so azospirillum is mostly uh, found in the rhizosphere of grasses. It is isolated from the rhizospheres of many grasses and cereals in tropical as well as temperate climates. Species are Azospirillum lipoforum, Brasiliensis, Amazonis, Halopreferens, Iracans. So, based on the area from it as the strain is isolated, the species name is given. Then it is because it has a single flagellum at the end, so it is motile. A Brasiliensis and A lipoforum and Iracans डिस्प्ले मिक्स्ड पैटर्न ऑफ फ्लैजिलेशन. किसी में एक फ्लैजिला होता है, किसी में एक से ज्यादा, किसी में दोनों एंड पे भी हो सकता है. ओके, बट मोस्टली एक एंड पे होता है. One polar flagellum is synthesized during the growth in the liquid medium and is primarily used for swimming. Additional lateral flagella are induced during growth on solidified media and used for swarming of the bacteria over the slide surfaces. A halopreferens and Amazonis only displays the polar flagellum. This exhibit positive chemotaxis. Chemotaxis क्या होता है? To move towards chemical stimulus. Chemotaxis means to move towards touch senses, touch stimulus. Then they show directed movement toward the optimal oxygen concentration called aerotaxis. So, chemotaxis, aerotaxis, chemotaxis. Phosphate solubilizing bacteria. Now we have phosphate solubilizing bacteria. So this will be your last topic. What are phosphate solubilizing bacteria? They solubilize the phosphate. They degrade the phosphate phosphate containing compounds in the soil, so that phosphate is released from it, and now it can be absorbed from the plant, uh, absorbed by the plant roots. So nitrogen fertilization is often not efficient enough to make the needs of plants growing, especially in calcareous and alkaline soil. Therefore, using phosphate solubilizing bacteria, that is PSB, for providing necessary phosphate is necessary. The conversion of insoluble phosphate compounds in the form accessible to plant, that is soluble form, is also done by PSB. In PSB constitute about 1 to 50% in the soil, uh, means total population, 1 to 50% of total population of bacteria in the soil. So they have been isolated like simple serial digestion method, and in so they are isolated from all areas, including rhizosphere, non-rhizosphere soil, rhizoplane, phyllosphere, and rock phosphate deposits. Rhizoplanes means surface of uh, root. Rhizosphere means after surface, from surface to some distance, like it can be 1 millimeter or half millimeter or even less. So that area is called rhizosphere. Rhizoplane is just surface. Phyllosphere is surface of leaf. The ability to solubilize insoluble organic phosphate and compounds such as hydroxyapatite. So I told earlier that phosphate is present in the compound form in some of the uh, in form of compound in the soil in some. So some example of those compounds are hydroxyapatite, tricalcium phosphate, rock phosphate, dicalcium phosphate. So what this PSB will break these chemicals and release the phosphate. Phosphate then molecule phosphate ion is then soluble in water, and it will be absorbed by the roots. It will after dissolving from the soil, it can go to the root surface by chance, by random association, and it is taken up by the root surface. So how does this break down this compound, all the bacteria, PSB or zinc solubilizing or potash solubilizing? So they have many mechanisms. So one is liberation of acids, which may be organic or inorganic acids. Second is liberation, secretion of some enzymes which do as catalyst for breaking down these uh, molecules or compounds, and secretion of siderophores, and secretion of some chemicals which will bind, which will have more affinity than this compound for phosphate. So example of PSB are Streptomyces, Agrobacterium. So there are many PSB like Agrobacterium, Azotobacter, Achromobacter, Acetobacter, Burkholderia. Then you have uh, Azospirillum, not Azospirillum, Bacillus. Bacillus, some species of Bacillus. So let us read some of them. Streptomyces, Agrobacterium, Azospirillum. Some species is also able to solubilize uh, phosphate beside the beside solubilizing the nitrogen. Then bacillus, different species of bacillus, megatherium is especially important, and pumilus. Then Klebsiella, Klebsiella, a species of Klebsiella is also used for uh, is also causing the pneumonia disease. Klebsiella pneumoniae. Then Vibrio, which causes the cholera. Different species Vibrio cholerae causes the cholera. So Vibrio proteolyticus is found in soil which break down the phosphate. Then Aerobacter aerogenes, Achromobacter, Enterobacter, Thiobacillus. Thiobacillus peroxidans is very important bacteria. This can degrade some heavy metals. This can solubilize nitrogen and phosphorus and other metals also. Uh, Not metals, these nutrients also. So it is very important bacteria. Then Citrobacter, Arthrobacter, Gordonia, Azotobacter, Pentonia, Nitrosomonas, Nitrobacter, etc. So, by mobilizing inorganic or organic phosphates, symbiotic nitrogen reservoir like Rhizobium leguminosarum and Rhizobium species, nodulating Crotalaria species also improve plant phosphate nutrition. Okay, so these are not very important for you. How this other methods of solubilizations are there. So, in briefly, I can tell what are these. Number one, immediate distribution of phosphate between available pool in soil and total soil phosphate. Convert this insoluble phosphate into available form through acidification, chelation, exchange reactions, release of complexing minerals, uh, complexing or mineral dissolving compounds which are organic acid anions, protons, hydroxyl ions, carbon dioxide, secretion of citrophore, oxygen production, ACC deaminase activity, release of organic acids or mineralized organic phosphates, enzyme catalysis through phosphatases, etc. So this is how they make this available to the plants. Okay, so I have told you about cyanobacteria as well as phosphate solubilizing bacteria. There are other bacteria also which are sold. For example, zinc solubilizing and potash solubilizing, iron solubilizing.